Bună dimineața! Se intonează imnul României. Distinguished Mr. President of the Senate, Professor Vasile Vântu, distinguished teaching staff and researchers from Romania and abroad, dear students, dear PhD students, dear colleagues. Today, at Iași University of Life Sciences, we are opening the 11th Congress entitled Life Sciences Today for Tomorrow. On behalf of the university management, we are welcoming all participants from 8 foreign countries: Cameroon, France, Germany, Italy, Serbia, Spain, Republic of Moldavia, and Republic of Turkey, representatives of universities, researcher institutes, and science academics. we greet the presence of the representative of the Universities of Agricultural Sciences and Veterinary Medicine from Bucharest and Cluj, as well as the presence of the representatives of the University of Life Sciences of Timișoara. We wish you may all enjoy your stay in Iași during and after the scientific dialogue within the 11 sections of the Congress, in which will be delivered 199 presentations and 173 posters in these 2 days dedicated to our Congress, with a total of 372 papers and 17 workshops. Please visit the Congress details, the program of the faculties and posters, scanning your badge. For young scientists, we will select by peer review around 10% of all presented papers to be submitted in prestigious journal with high impact factor accepted by different houses, printing house like Elsevier and other, all texts being supported by organizing committee. So good luck and pay attention. I have also the pleasure to thank to those keynote speakers who accepted to have a plenary presentation, and please let me introduce them with a very short characterization, invited in order by faculties of agriculture, horticulture, animal husbandry, and vet medicine as follows. We have in front of us the first presenter, Professor Patrick Scheffer, who will present us here RNA interference and priming in crop protection. So a few words about Patrick Scheffer. Let me not present the entire curriculum vitae of these personalities, because we have not so long time. You can find them on different sites. So I just have a few words. He was director and professor at the Department of Phytopathology in Justus Liebig University in Gießen, Germany, since 2022. Professor in molecular botany at Ulm University, Germany. Honorary professor, fellow, School of Life Sciences, University of Warwick in United Kingdom. Principal investigator at the Warwick Integrative Synthetic Biology Center from United Kingdom. Associate professor, School of Life Sciences, University of Warwick. Habilitation in and Venia Legendi for Phytopathology and Cell Biology. Research group leader, Justus Liebig University in Gießen, Germany. And postdoctoral research associate at Justus Liebig University, Gießen, together with Mr. Kogel. So the second presentation, named Circular Economy and Life Science, a continuous and valuable dialogue, will be presented by Professor or Professoressa Alessandra Bognoli, PhD. She is full professor at the University of Bologna. Her main research topics are in environmental engineering, with particular regard to treatment, recovery, and recycle of raw materials and solid waste, to the life cycle assessment and circular economy, to dissemination of urban green technologies, and unconventional building materials for climate change resilience in urban areas. She founded and coordinated the research group of Transition Engineering, whose activities are aimed at building a sustainable future by environmentally, socially, and economically point of view through conservation and valorization of natural resources, raw materials, water, and energy. A precise research concerns the application of above topics in cooperation with cooperation activity for developing and emerging countries, numerous publications in collaboration with national and international universities. It's delegate of UNIBO at UN Sustainable Development Solution Network and at UE Commission Operational Group of the European Innovation Partnership on Raw Materials. The third presentation here in plenary will be, it's named Understanding Eggshell Formation to Maintain its Quality in Lying Hens, and presenter is Joel Gautron, Alejandro Rodriguez Navarro Miquel Duclos. He works at INRA, UMR BOA, Université de Tours, Neuilly, France, Departamento de Mineralogia e Petrologia, Universidad de Granada. Research Director on National Institute of Research for Agriculture, Food and Environment in France for 14 years, specialist in egg biology and biomineralization, scientist in avian biology and poultry, and research unit in collaboration with the University of Tours. Focuses on integrated research in the biology of birds from the molecular level to that of the animal in its environment. Knowledge in this field of psychology and genetics that contribute to the development of sustainable livestock systems. The fourth presentation, named Competence Center for Climate Change Digital Twin Earth for Forecast and Societal Redressment, abbreviation is DTE Climate. Presenter is Professor Mihai Datcu from the National University of Science and Technology, Politehnica Bucharest, Romania. His interests are in Bayesian inference, information and complexity theory, stochastic processes, model-based, scene understanding, image information extraction for application in information retrieval, and understanding high resolution and optical observation. He has held visiting professorship at the University of Oviedo in Spain, the Louis Pasteur University, and the International Space University, both in Strasbourg, France, the University of Siegen, Germany, and the University of Innsbruck, Austria, the University of Alcalá, Spain, the Tor Vergata University, Rome, Italy, Universidad Pontificia de Salamanca, Madrid Campus, Spain, University of Camerino, Italy, Swiss Center for Scientific Computing, Mano, Switzerland, so and so and so. He was full professor of DLR CNS Department of Paris, Paris Institute of Technology, Telecom Paris. He initiated in European project framework for image information mining, and it's involved in research program for information extraction, data mining, and knowledge discovery and data understanding with the European Space Agency, ESA, NASA, and in a variety of projects, national and national, European projects. He is a member of ESA Big Data from Space Working Group. Now, as invited, it's also the director of National Competence Center, financed by Resilience National Program and titled shortly DTE Climate. The fifth presentation, it's the stereotypes and the influence on cognitive performance. And presenter will be our colleague, Associate Professor Dr. Șerban Moroșan, who actually working at UMS 28 Sorbonne University at INSERM, Paris, France, Department of Public Health, Ion Ionescu de la Brad-Iași as Associate Professor. Șerban Moroșan is the Director of Biological Research Facilities at Sorbonne University, Paris, France. He represents Sorbonne University at League of European Research Universities. He represents INSERM, the French National Institute of Medical Research in Europe, to the European Commission and European Parliament. He is also an associate professor at the Faculty of Veterinary Medicine in Iași, Romania. He earned his PhD from Angers University, France, with a specialization in biomedical sciences. Dr. Moroșan has an extensive background in translational medicine and laboratory animal science, where his work focuses on advanced phenotyping technique and preclinical research. His contributions have significantly advanced the understanding of disease mechanisms and therapeutic development using small animal models. So I introduced briefly our presenters, and now thank you for your attention. I give the floor on the first presentation to Mr. Patrick Scheffer. Please, Patrick.
So, first of all, welcome all, and it's really a pleasure to be here and also an honor to be here, the first speaker of the Congress. And really thanks a lot to the invitation and also hospitality of the university. Really a pleasure to be here. And first time for me to be in Iași, and so far I really enjoy to be here. So, as already mentioned, my talk will be about my field, which is plant disease and plant protection against disease, obviously. And the motivation of my work and of my talk is obviously that we have to find new ways to protect plants, especially in the prospect that we need to reduce pesticide application, but at the same time provide farmers with solutions that can really protect crops against diseases. So my talk will be about RNA interference and priming as strategies. So I will present two projects that are actually, one is funded by the Ministry of Agriculture and Nutrition in Germany, and that is called primed wheat. So just a brief introduction, because probably not everyone is really familiar with the concept of plant protection. So what you see here is actually the situation that plants face. So most of the time, plants are actually resistant against microorganisms, because microbes in general that are in the soil or colonizing plants are not really able to infect plants and cause diseases. And during evolution, some of these microbes have specialized. Some of them became pathogens, and others became beneficial symbionts. Both of these groups of microbes, they have the same task. They have to breach barriers, overcome plant immunity to colonize plants at the end. And they have to manipulate the plant in order to get nutrients, because that is what they want. However, the outcome of this is very different. In case of pathogens, we see a disease. In the case of beneficial microbes, we see that these plants are becoming more resistant. So if you think about beneficial symbiosis, they are quite old in principle, because when plants colonized land more than 400 million years ago, they were already associated with microbes, with beneficial microbes. And the reason that they could actually colonize land was that these microbes at that time already provided protection, and they provide nutrients to these plants. And only with this protection and with this support, plants were able to colonize land. And I just have here at the bottom, you see the temperature and the carbon dioxide concentrations 400 million years ago. And you see the average temperature in comparison what we have today on Earth was far higher. And actually this is like a catastrophe. If we would think nowadays, we would live under such a climate and under such an environment. So, but indicating that the potential actually that beneficial microbes have to protect plants. So, and as I said, what these beneficial microbes help plants with is nutrient supply. They help plants to develop roots actually from the time of land colonization till today, and they protect against stress. And the question is, of course, can we access these benefits for crop protection? So the fungi we are working with are listed here. We are mostly working with one fungus that is called Serendipita indica, which we can see there. We can culture on plates. And the nice thing about this fungus is it has a very broad host range. So it colonizes all crops, and to all these crops it more or less gives the same benefits. That is one you see in the bottom, growth and yield promotion. When the fungus colonizes plants, it gives abiotic stress tolerance, here in this case drought, but also against salinity. And you see also that these plants are more resistant against pathogens. One has to say this fungus only colonizes roots, but the whole plant is protected. Especially leaves are also protected. And we call this phenomenon priming. So the idea is, of course, can we somehow use and access this agricultural potential of this Sebaceanium fungus that I listed here. So priming, what is priming? At the end, what you see here is a plant colonized, or plant colonized by some bacteria, fungi, or mycetes. This interaction, if they are beneficial microbes, they can induce systemic signaling, and then the whole plant is resistant against pathogens. So, and as I said, this is a phenomenon that we call priming. So priming is then a physiological state of enhanced alert. That means as soon as these plants are more or less prepared for a pathogen attack, and once a pathogen will colonize these, or tries to colonize these plants, the plant can faster and more robustly respond with a defense response. Part of this is also that we see increased biomass, so yield. And what we want to do in this project is we want to evaluate the genetic variability in wheat for this priming, and identify also genomic regions and genetic traits that are involved in this priming by genome-wide association studies. So what we did is more or less, we had about 208 genotypes. This was an approach in collaboration with the Julius Kühn Institute in Germany. And these 208 genotypes plus 50 genotypes from breeders, we just inoculate these plants and then just checked and phenotyped how good is priming in these plants. And what we phenotyped against is one's powdery mildew disease as a leaf pathogen, and we looked for root and shoot biomass if this was increased. And our colleagues did something similar with another beneficial bacteria, and they looked for rust phenotypes. At the end, we came up with screening all these genotypes. We got data in both sets for about 170 genotypes that we could use for GWAS. So, and what was quite interesting, because in the textbook is still today, people say, or is stated, that priming is not genetically determined, but actually what we see, it's clearly genetically determined. So first of all, you see here some pot experiments where whenever S. indica is present, we see that the plants grow better, and they have a far better root development. And on the bottom, you see a bar chart, and these are not all the genotypes, just a representative set. And you can see from the bars on the left-hand side that some of these genotypes, they respond worse when the beneficial microbe is there, and we see some that perform far better. And the same is on the right-hand side for powdery mildew infection. So, and we obviously are mostly interested in those that show with the beneficial fungus enhanced biomass and enhanced resistance. And this is just an example of two wheat varieties, Contra and Congo, and you see with the red line, that is actually S. indica, and in both cases, S. indica over the time shows less disease development of the powdery mildew. And in green, you see also a beneficial bacteria, where we also see improved performance of these genotypes. And then obviously what we do is we do GWAS with these 175 genotypes to identify regions that are responsible responsible for this enhanced resistance. And then at the moment we have now QTLs identified, and these are then the genetic traits that we want in future to functionally analyze and also see if we can transfer them to elite varieties in wheat. In addition, we do field experiments. We have 2 breeders involved in this project. This is Limacrin and AHT from France. So we have 3 field sites together with Julius Kühn Institute. At the moment, based on our initial screen, we have now 30 genotypes that we test in the field. We do also now seed coding of S. indica so that it is more easy to apply, and the farmers would accept this then as well. And we do the feed evaluation that we obviously check for different developmental stages and disease and yield. And I think especially in April 2020, we could nicely see also in the field that S. indica inoculated plants perform far better. This was actually also in 23 visible when you were in the field. On the picture it is less obvious. So this is quite promising, and the breeders are really excited about this and see this as an opportunity to have new traits included in elite varieties. So the second project I want to talk about, and this is funded by the Federal Ministry of Education and Research. This project is called EPIC Wheat Interfere, and this is about non-coding RNAs. And just a brief overview, so if you look in plants, but also in humans and animals, we have a number of non-coding RNAs. So non-coding means, of course, these RNAs are not translated into proteins. Some of them, they have a housekeeping function that you see here on the left-hand side. They are involved in translation, splicing, modifications of mRNAs, and others have a regulatory function. And so they are involved in to what extent mRNAs are transcribed and to what extent they are also translated into proteins. So just 2 weeks ago, actually, Rafkin and Ambrose, they got the Nobel Prize for in physiology and medicine, and for their discovery of microRNAs as really regulatory small RNAs and their role in gene regulation. This was now the second time that actually in this field there was a Nobel Prize. In 2006 there were Fire and Mello who got the Nobel Prize for RNA interference. So indicating that these non-coding RNAs are quite important for physiological and also morphological functions of organisms. So what is RNA interference? This is just depicted here. So we have regions in the genome that code for non-coding RNAs like double-stranded RNAs, and these double-stranded RNAs are actually processed by a protein called dicer, and then we get short or small RNAs. They have a length of, let's say, 20 nucleotides. These are then further processed, and together with Argonaut, there is a RISC complex formed with a single strand of this small RNA, and then based on complementarity, this small RNA finds matching mRNAs, and these matching mRNAs are then silenced by slicing, so they are simply cut into pieces. So this process is obviously highly specific because these small RNAs only bind to those mRNAs that really have a sequence homology. What is quite interesting, and this is a concept approved more than 10 years ago, that plants and microbes exchange these small RNAs between each other. So if you see there in blue, these are pathogens. They synthesize such small RNAs or small interfering RNAs and bring them into the plant cell. And these small RNAs have, of course, a function in the plant to find those matching mRNAs and integrate them. And the mRNAs that they target are mRNAs involved in plant defense. So it is just a mechanism how pathogens try to overcome immunity and manipulate the host plant. And plants do the same thing. They synthesize small RNAs, bring it into the pathogen, and these are then small RNAs that try to find mRNAs that are involved in pathogenicity. And we are in the project really interested to find those small RNAs that plants are producing that we think have a potential to serve as kind of biopesticides in future. So that is the whole idea. The innovation is use RNA interference in crop protection. And we at the moment work on fungi or pathogens, but it can be used against all sorts of pests and diseases, including viruses. So a proof of concept looks a bit complicated, but at the end what it shows is you see on the right-hand side, these were some constructs that were designed against this RNA interference machinery. So we designed small RNAs, or Bernhard Werner in my group designed small RNAs against these argonauts and dicer-like proteins or genes, and then these are argonauts and dicer of Fusarium graminearum. And when he sprayed these small RNAs targeting this Fusarium graminearum argonauts and dicers, you see in the bar chart the disease level. You always see as soon as we have a small RNA against these genes, you see that the fungus is not so able to colonize the plant. So, and it is a nice proof of concept because it shows us also that we can spray small RNAs on leaves that are colonized by the fungus and stop with this the fungus. So what we do is, although it's called wheat interfere, we're not working with wheat because this is a big sequencing approach and wheat would be quite complicated. Instead, at the moment, we use a model for wheat, which is Brachypodium distachyon, and this is then inoculated either with Fusarium, then with Puricularia, which is formerly called Magna Porte oryzae, and the rust fungus. And then we do so the project outline is then that we want to identify which mRNAs are actually produced during this interaction, then which small RNAs are actually produced, and we do a decadome sequencing. We also want to identify that we see which mRNAs are actually degraded. So the idea is, of course, we find a small RNA that is produced in the interaction, we find the matching mRNA that is most likely then downregulated, and at the same time, and that is for us a proof then that the respective mRNA is then also degraded. So just very briefly, we get, of course, a lot of mRNAs and sRNAs in our sequencing approach. We see some overlap, but not much in the RNAs, mRNAs. What is quite interesting is if you look at the sRNAs that plants produce in the different interactions, they are different. So the plant has somehow regulation and recognition that it produces different small RNAs if it is Fusarium or Puricularia, for instance. I have not included rust data because this is at the moment sequenced, and we will get within the next weeks actually the data for this as well. So that is a bit complicated. What you see here is more or less the decadome sequencing readout on the left-hand side. The sequence that you see and what is underlined there is actually the site where we see a silencing or a slicing. So where we see that the RNA machinery cuts the mRNA into pieces. With this, with the chart next to it, it indicates from which organism we find small RNAs of 20 nucleotide length, and at which position actually the nucleotide is where we see a slicing. And that is quite important to see because there's a certain dogma about the starting nucleotide of the slicing, and we see really that there is a specificity of the interaction, how certain small RNAs perform slicing. And if we take all our data together, we see that actually the plant releases 9 small RNAs in the case of Puricularia and 94 for Fusarium. This sounds quite small, but we are very, very stringent in our analysis because we want just to see those small RNAs that perform the best. And we see, of course, also small RNAs coming from the fungus and injected into the plant. So therefore our approach looks really, really promising. So we see really sRNAs that target mRNAs specifically. We see that these target mRNAs are downregulated, and with our data we see that these respective mRNAs are also sliced and with this silenced. So the question is, of course, what we see on top. Very briefly is, there is a certain debate on which small RNAs are actually active, and it's often said that it's only those small RNAs that have a certain length of 19 or 20 nucleotides, but we see actually a far bigger portion of small RNAs of different lengths that have a silencing activity. So can we validate this data? We developed a method to also see the slicing of small RNAs and independently approve this. We call this method nil-tedious, which is just, without going into detail, indicating really in a quite short time if slicing of a given sRNA is really happening in planta. So here is just shown a decadome sequencing readout, and you see these dots there. And the more dots you have, and the higher the piles of these dots, then we see there's a slicing event happening in the gene. So at the bottom axis, you always see an mRNA in its full length in a different position, and you see there around position 1400, there is actually a small RNA from Brachypodium that slices this gene of the mRNA of the fungus at this position. And below this is our nil-tedious readout, where we just check this small I has really a slicing activity, confirm this data, and you see also there dots at the right position where we see slicing. So then obviously when we have our candidates, and that is shown here in the bar chart, we have two controls, MOC and GFP, so we have small RNAs against GFP as a control, and we have two candidates, and you see for instance this small RNA against Fusarium graminearum gene 16. We see when we spray this small RNA, the Fusarium is colonizing or has difficulty colonize plants. We have a second candidate, and that did not perform that well. But if we looked actually and checked again how this slicing is, you see on top what I showed you before for this candidate 16 and below the candidate 23. We saw also a cloud but in the decadome sequencing data, but it was not too obvious. And we look at nil-TDS, actually there was not really a slicing activity, and which just confirms that this candidate 23 has not such a high activity and therefore no phenotype. So to sum it all up, we have obviously done genotype screening for priming, identified really candidates and genetic traits that we think contribute to priming in wheat. We have the same data set also for barley, and we can now compare these data sets and hope with this we find kind of conserved genetic regions that we can now use in breeding programs. With the non-coding RNAs, I think our method is quite clear that we can identify small RNAs and their activity, and that we can use these small RNAs in future to spray them and to control diseases, and that we, with this, have a new concept in crop protection. So at the end, I just want to acknowledge, of course, those people involved. The Primed Wheat project was actually started by the previous head of the department, Professor Kogel. When he retired, he was just asking, Patrick, you like to continue this project? And he did actually all the groundwork. And I said, of course, I want to continue because it's, I think, a quite exciting project. People mostly involved from our side is Jenny Thielmann as a PhD student and a technician, Ute Miknas. We have really, really good collaborators at the US Kuhn Institute and obviously at the breeders. And then we have Wheat Interfere. And there are special things, especially to Bernhard Werner, who is a postdoc leading the project, and Rebecca Schmidt as a technician. And here we have, with another Julius Kuhn Institute, also a very, very nice collaboration. We thank, of course, the funders for this. And just to announce that we have open PhD position. If someone in the audience is interested, just get in touch. And maybe in future you can do a PhD at my lab in Germany. Thanks a lot for your attention.
Thank you, Professor Patrick Schäfer from Justus Liebig University Gießen. So from specialists from this hall, if anyone, yes, you have questions, yes, please, because all is registered here.
Thank you, Patrick. Really nice talk. I have two questions. Actually, I have lots of questions, but two major ones. So first one will be on how scalable are both systems for a farm level? Would it be possible at some point to say with a small cost you can prime the seeds, or you can spray with RNAs and then have farmers be able to pay this? That would be the first question. And the second question that I have is for the small RNAs, did you check if it interacts with other organisms in the microbiome and positive or negative, or not only the pathogen? Great, it works with the pathogen, but the rest of the microbiome?
Yeah, very good questions. Yes, the priming is obviously something that we hope we can introduce then in future into elite varieties. I have also to say some of the elite varieties show also priming, which indicates us that these traits are not necessarily present in all the elite varieties. So it is scalable, and we hope in future we can introduce this. How fast that goes is, of course, always a question, and I think breeders in the audience know how long it takes sometimes to get such traits into plants. But we think it's feasible because the breeders are very optimistic about this. For the small RNAs, obviously we can design small RNAs with high specificity, also artificial ones. We have not checked side effects. That is something we really want to do in future, because that will be very crucial to get acceptance and also approval to apply small RNAs in future. That is a very, very important point we have to look at. So it's still quite a way to go before we can really apply small RNAs in the field, but we believe that can really be a game changer if we manage this. Because we can, for instance, also design 5, 10, 20 small RNAs against a given pathogen and really knock it out completely. Yeah.
Thank you. Anyone? No? Thank you so very much, Patrick, for your presentation. Thank you very much. Now the second presentation is from Professor Alessandra Bognoli from the University of Bologna. We expect your presentation named Circular Economy and Life Sciences, a continuous and valuable dialogue. Thank you.
Good morning, everyone. Thank you very much for your kind invitation and a nice presentation. I would like to talk about, let's say, a more general topic related to circular economy and life science, a continuous and valuable dialogue in which way they can collaborate and to find solution for the ecological crisis we are living in. Just to start, the context we are living in. In fact, ecological crisis, climate change, resources depletion, dramatic resources depletion, both for water, raw materials, and energy sources, and generally speaking, the pressure on Earth's life supporting system. Just to show you a couple of graphs showing what happened on our planet since the human life, the beginning of human life, we can recognize for any socio-economic trend a sort of similar behavior. Since the beginning of our history, a constant level or a slow growing during the last century. But after the Second World War, basically on the 50s of the last century, a dramatic growing some scientists define that period the great acceleration period, looking at each of socioeconomic trends, just looking at the world population or the primary energy use, the water use, the fertilizer consumption are dramatically growing. And the same behavior looking at Earth system indicators. One of them, for instance, carbon dioxide concentration in the atmosphere, or the surface temperature, or the biodiversity loss. The same attitude, just to show how dramatic is the human footprint on our planet. Some scientists define the period that we are living in as Anthropocene, as you know, the current geological era in which the terrestrial environment in all characteristics is influenced both locally and globally by the effects of human action. We have to find solutions for sure, and I suggest some possibilities related to the interaction between circular economy and life sciences. Another model, the so-called down-out economics, really fascinating, shows as our planet is characterized by ecological ceiling, and unfortunately during the last few years we exceed the ecological ceiling. For instance, the climate change, the biosphere integrity, the freshwater quality and quantity, and some novel entities that can have large-scale impacts on Earth ecosystems. So, in very short, we can. I would like to remind you what circular economy means, but that is the most common representation according to Ellen MacArthur Foundation definition. We can see that several cycles can be closed in order to guarantee a proper raw materials, energy, water consumption, to avoid so much waste in the environment. And several loops can be closed, both talking about, generally speaking, raw materials in the so-called technical material cycles, but I would like today to focus on the biological materials, because the interconnection between circular economy and life sciences. All organic waste coming from any kind of activity, from agriculture to our common life, can be valorized in several ways in order to close the loop once again and to save natural resources. The circular economy represents one of the main pillars in the European Green Deal. In fact, in March 2020, the European Commission adopted the new Circular Economy Action Plan, involving several, let's say, raw materials and waste streams. But in particular, I would like to focus on some specific topics, such as packaging, plastics, food and water and nutrients. And these are the topics inside the interconnection between circular economy and life sciences. Just some examples of that relationship, right? So we can talk about the contribution of life sciences in climate change, in general in climate change related issues. It is possible to develop technologies that are able to reduce carbon emissions and contribute to carbon capture and storage. basically the two solutions that can mitigate climate change. How is possible? By using microorganisms or plants that are able to sequester carbon dioxide from the atmosphere. The other topic so interesting today is the production of bioenergies, generally speaking, coming from waste, from organic waste, from biomasses that are able to produce this kind of energy. Here, just an example related to wastewater treatment, able to support algae cultivation, and thanks to a process, they are able to produce bioenergies. The same in a natural way, coming from algae bloom. But really interesting also the bio-based materials topic. You know, bioplastics is quite a new frontier in new plastics production, coming from biomasses and organic waste. Bioplastic can drive the evolution of plastic, at least for some application. In my opinion, the best application is in the food packaging, for instance, because the quality of this kind of plastics. Then in the sustainable food system, the life sciences contribute to more sustainable circular models by minimizing resource use, such as water, fertilizer, pesticides, valorizing of waste. Another interesting field of interest, the healthcare and pharmaceuticals. In order to promote, circular economy model is able to promote new way in the, for instance, medical devices or packaging in that sector managing. In fact, we have to avoid at all single-use device, for instance, and to promote the recycling of several ingredients, or in general several materials coming from this kind of sector. A very interesting perspective in the agromining. It is possible to extract, to exploit metals and other minerals from plants. The so-called biorecovery of metals. In that way, we apply directly a circular economy by using plants for the metal extraction. Basically, the microorganisms are able to convert the solid chemical into a soluble substance, and we are able to recover metals by using engineered microbes. But the last topic could be wastewater treatment. Wastewater treatment, recycling and reuse. We are experiencing a dramatic water scarcity around the world, once again because of climate change effects, but also because of the growing in the number of people related to the worldwide population growing, and so we have to find several solutions to implement water resources. Wastewater recycling could be an answer. We know that it's necessary for sure to properly treat wastewater, because otherwise several issues in terms of humans and environmental health risk. But wastewater treatment could be also an opportunity to recover water, at least for some purposes. For instance, benefits for agricultural purposes. Wastewater reuse in agricultural irrigation, for instance, thanks that we are able to save water, generally speaking, to use less chemical fertilizer, and to manage at best in general the wastewater. Main uses both in non-food crops or food crops. Some very interesting natural systems for wastewater treatment. You know perfectly what phytoremediation and aerated lagoon mean. Phytodepuration is a natural treatment that reproduces the natural purification processes in a controlled environment. We are talking about artificial little deep basins filled with inert material and fed with aquatic plants, basically macrophytes, that are able to filter the contaminants and to reduce the pollutants inside the water. The aerated lagoon combined with the phytoremediation system could represent the final treatment stage and the natural disinfection. They can be applied for any kind of wastewater treatment. In particular, considering the urban wastewater, or generally speaking, wastewater treatment, the last, this year, European directive suggests to move in three main directions, such as the water quality, for sure, and it is necessary to remove all nutrients, and the new treatment of micropollutants, and to test the water in terms to avoid new pollutants inside the water. The Green Deal is suggesting the possibility to reuse and recycle water, wastewater, sludge, and nutrients, again, such as phosphorus. And by governance point of view, a couple of subjects are really interesting in that subject, that is the extended producer responsibility enlarged to cosmetics and pharmaceuticals and many other product systems, and health parameters monitoring. The new pollutants, such as, for instance, antibiotics or other pharmaceuticals in the urban water. Thank you very much for your attention, and mulțumesc, everyone.
Thank you. Thank you very much for your presentation. And now, question if you want to address. Okay, I saw.
Thank you very much, Professor Bonolli. You have presented this wonderful lecture for us about circular economy. How do you see the applications of circular economy in horticultural and landscaping technologies?
Thank you very much for your question. That is an important question. Basically, I think we can apply circular economy perspective, generally speaking, as a first step in saving natural resources, saving the use of several additives such as pesticides, fertilizer, and so on. After the saving, the prevention of the utilization of several materials and products, generally speaking, there's the possibility to promote recycling. Circular economy doesn't mean just recycling, right? So we have several possibilities to promote circular economy, but finally the recycling could be the best option for several applications. In this case, it's necessary to promote recycling of several organic waste, biomasses, several residues coming from horticulture and agriculture or land use, generally speaking. So I think it's possible to promote recycling in order to avoid the waste production from one side and to reduce natural resources supply, exploitation, and finally depletion of them. Thank you. Yes, another question?
Thank you very much, dear Professor, for interesting presentation. My question is, do you think there are barriers to make more easily or a little bit fast the transition from the linear economy to the circular one? And if yes, which there are? Thank you so much.
Thank you very much. Nice question, and you're totally right, because several, let's say, inertial attitude to move from the linear to the circular economy. What I commonly say is that we have to demonstrate that circular economy is effective not only by environmental point of view, by natural resources depletion issues, waste production and so on, but also by economical point of view. For sure, also considering the social impact that is, commonly speaking, is effective, because we are able to create, for instance, new jobs and new job positions, but also by economical point of view. If you are able to consider, let's say, a sort of life cycle also in terms of economical aspects, we can demonstrate that is more convenient, also by economical point of view. Thank you very much. Yeah, another question?
Thank you for your nice talk. I'm interested in your perspective over the term of bioplastics. What do you mean by bioplastics? I'm dealing with polymers, and there are the polymers that are natural polymers that are degraded, naturally degraded, or there are plastics that, like polypropylene or polyethylene, that may be degraded by some microorganisms. So what's the meaning you're hitting?
Thank you very much. Basically, bioplastics are produced not by using fossil fuel or fossil resources, but organic-based materials. And basically, I appreciate the production of bioplastics from biomasses and organic waste. I think that a huge production in bioplastics is not possible. We cannot substitute all fossil fuel-based plastic, fossil-based plastics with bioplastics, because there is the production of bioplastic can fight the food production, for instance. No, there are no enough lands able to produce bioplastics. I don't like them very much, but generally speaking, bioplastic are confused with biodegradable plastics or composting plastics. That is another subject. Maybe some plastics can become biodegradable, can be biodegradable or compostable, and can be managed together with organic waste. That's the reason why I suggested that, in my opinion, the best solution to use bioplastic is to substitute packaging for food conservation, for food packaging. Just for that specific application, bioplastic or biodegradable or composting plastics can be effective, not generally speaking. Talking about plastic, I would like to highlight the fact that we have to reduce plastic consumption, or better, to manage at best plastic waste. A substitution, a partial substitution, a very little percentage substitution can happen, in my opinion, just for that application. Thank you. Thank you very much.
Thank you, Professor Alessandra Bonioli. And now the third presentation is from France. Yes, it's coming. Joël Gautron.
Thank you. Thank you, Mr. Chairman. Good morning, ladies and gentlemen. First, I would like to thank the organizer and specifically Professor Razvan Radu for their kind invitation. This is my first time in Romania. I'm very pleased to be here today. And I'm going to speak on very important animal production, which is the egg production, that you know is a basic ingredient for human food. And I will focus my talk about the eggshell, that is an important structure of the egg for the marketing of the egg. Okay, so let me give you some socio-economic context data. Okay, just put a laser pointer. You know that the original function of an egg is not to be eaten. It's just to be an autonomous closed chamber that allows the embryonic development. And you have to imagine that from the day 0, when you have an egg, from day 21, when you have a chick that can go out the egg, you have no possibility to put something inside the egg. And you will not have any possibility to put something outside the eggs. That means that the egg is relatively remarkable because it must contain all the entire components necessary for life. And that means that these components are well-balanced nutrition ingredients, highly digestibility, highly value. The 100 references for World Health Organization of the balance of amino acids in the egg white, for example. And there is a lot of compounds, more than 1,000, with a broad range of biological activities. And of course, this is the reason why we are eating some eggs. Just a parenthesis, for the table eggs, there is no fertilization, of course. The birds do not need to be fertilized to produce eggs. And also, if we have this development outside, in the external milieu, the young chick has to face some stress, the physical stress, but also some microbes that want to enter in the eggs and go to the yolk to eat something. So there is also some protective system that we call the natural defenses, which are mainly the shell, which produce the physical defense, and some proteins and antimicrobial proteins which are present in all compartments of the eggs, and they are named the chemical defense of the egg. Okay, so the table eggs, this is a basic ingredient for human food. This is the cheapest animal food of high nutritional quality. Also, this is a food with no religious prohibitions. And about 70 million of tons of eggs are produced each year in the world. That represent more than 1,400 billion of eggs each year. Only in France, my country, I do not have the number for Romania, I'm sorry, but in France we produce 15 billion of eggs in France each year, only for human consumption. Okay, eggshell, why do I speak from eggshell? Eggshell, is not more interesting because it's the only non-consumable part of an egg. But its quality is really crucial for the marketing of the egg, for different reasons. If you have downgraded egg due to deteriorated egg quality, mainly is due to dirty, cracked, or broken shell. So this is an economic issue. In the point of view of health issue, the eggshell as a physical barrier has to be very important and very efficient to avoid any penetration of bacteria inside the egg. So this is a main concern for health issue. And also I would like to focus my talk on ethical issue. We have a strong evolution of housing system and societal demand in Europe. The consumer demand for non-cage free-range systems. And in such system we have a lower shell quality. And also we have a strong demand for some sustainability reasons to have long life cycle and shell quality decrease with the age of birds. And for all this issue, the maintenance and improvement of shell quality is really important. And when we speak about shell quality, we speak about mechanical property of the shell. Just some few details. You can see on this slide for 2003, 2030, that the cages system has been changed in favor of alternative system that's been grown, organic production of free-range systems. And 80% of eggs were produced in cage 20 years ago, 50% in Europe 10 years ago, and now is less than 30%. In France, 5 years ago, we had 50% of eggs produced in cage, 12% in organic, and more than 30% in free-range system, including the label rouge with a specific French specificity. And today in France, the cage system representing only 1/4 of the total of eggs produced each year. So this is a strong evolution due to the social demand. Concerning the persistency of laying hens, we have breeding companies that have claimed that they will have developed the long life layer. They are capable to produce more than 500 eggs in production cycles. And you have the blue curves to show you what was, this is the egg production, the percentage of marketable eggs depending on the weeks of production, and the blue lines corresponding to the layers we had 20 years ago. And the red lines corresponding to what we have today. And 65% is the limit of marketable eggs for the beneficial limit for the producer. And so we can have some eggs coming from more elderly animals. But there is a problem, the maintenance of the egg and bone quality in such layers. You can say me, why to produce some so aged eggs? But only for sustainability. Even if a layer is laying 24 more eggs per hen, you reduce the numbers of layers in the world from millions in Europe. And this is an important concern. You know that now in France and Germany it's forbidden to kill the male from layers at birth, for example. So we have to find a solution to reduce the numbers of animals. Okay, let me talk now about the eggshell formation. Eggshell formation, this is a phenomenon of biomineralization. Biomineralization is a very important phenomenon on the Earth. It's allowed to reduce the level of carbonic dioxide on the primitive Earth, for example. And eggshell biomineralization occurs in the uterus, and this is the fastest process of biomineralization of the living life on the Earth. You have 5 to 6 grams of mineral, which is calcium carbonate, which are deposited within a 20-hour period. From inside to outside, you have here the egg white, you have here the atmosphere. You will find first, the first layer will be the eggshell membranes that you can see easily when you crack an egg, for example. These eggshell membranes are protein fibers, and they will support some organic cores, which are known as mammillary knobs. And these mammillary knobs are the seeding site where the first calcium carbonate crystal will form. And this calcium carbonate crystal form will grow, not in the direction of the membrane, because there is not sufficient space for competitive growth, but they will give some cones, and these cones, when they come close, they fuse to give a compact palisade layers. And these compact layers, sometimes you have no fusion of the cone, and it gives the pores. The pores are essential to allow the exchange of gas from exterior to inside of the egg, and they are essential for embryonic development. And finally, the most external layer is a bioorganic layer that is called the cuticle. It's very thin, but very important, because it comes to plug the pores to avoid the bacteria to penetrate. And soon as the egg is laid, this cuticle will dry, and it will make some small cracks to allow the gas exchange from the inside to outside, but it will not allow the penetration of bacteria in an egg. This is the reason why in Europe it's forbidden to wash the egg, to avoid to remove this very important layer. Some additional scanning electronic microphotography of the eggshell. You can see here the first events of mineralization. You can see a transversal section of the knobs, which are covered by the calcium carbonate, and you have all the adjacent cones. You can see here, you can have a zoom in this part, and you can see that there is a fusion, and sometimes there is no fusion, and it makes a gas pore. Concerning the cuticle, you can see it's a very thin layer, less than 20 microns on the surface of the shell. And if you look at the surface, it's cracked, like a dry soil, for example, in summer. And the nano fracture are at nanometers, so it will not allow the bacteria to penetrate into the egg. So the eggshell formation, let me give you some few details about the eggshell formation. The eggshell formation occurs in the uterus of a layer. And there is two main phenomena. First, the supplies of mineral for shell mineralization. It's really important in the layer. Why? Because the layer is only an animal of about 2 kilograms. It's very little birds. But to make a shell, you need more than 2.5 grams of calcium per day. So this is an important calcium metabolism. For comparison, for example, humans use only half a gram of calcium, and the average weight is 60, 70 kilograms. So this is a strong calcium metabolism, really exacerbated. Okay, this is a big metabolic effort. And when you have sufficient of calcium and carbonate in the forming milieu, you have the biomineralization process that will occur in different steps. The first low-speed step, the initiation of the first deposition of crystals, and the linear growth rapid phase, and finally an arrest of the calcification during the terminal phase and the deposition of the cuticle. On this diagram, you have all the organs, the most important organ involved in the calcium metabolism during shell form calcification. The oviduct, and mainly the uterus, the liver, we synthesize lipoprotein from the yolk, for example, the gut, the kidney, and the bones. The calcium that will be deposited in the shell will come for 100% from the diet. But there is a discrepancy between the timing of calcification and the possibility for feeding for birds. The calcification of the shell occurs at night, and it's only fed during the day. So there will be a deposition of calcium in a specific structure of the bone, that is a medullary bone, that will be solubilized during the night to give calcium for the uterus and to perform the calcification. So you have a natural osteoporosis of the bone. So this is a big scientific field of research, the osteoporosis on the bird, to learn from that. And of course, it's really dependent on vitamin D metabolisms. The vitamin D comes from the food. It's hydroxylate first in the liver in 25-OHD3. and secondly in the kidney to the active metabolite 1,25-OH2D3 calcitriol that will allow the transfer of calcium from different organs like the gut, the bone, but also we demonstrated in our lab recently that it was also strongly involved in the regulation of the transfer calcium in the uterus. Okay, if we want to have a strong shell, we need to transfer calcium, to combine the calcium with bicarbonate to give the calcium carbonate with one proton excess that will have to be removed, and it will be cemented by organic matrix, which are proteins and proteoglycans that give the shell. The calcium will have to come from the blood, will have to pass through the uterine cells and go to uterine fluid. And during this passage into the cells, we have to avoid to modify the calcium content of the cell because it's something that the cell do not support. So the control of calcium is very important. And also the same for bicarbonate, and the organic matrix will come from uterine cells and will be deposited in the forming milieu to form the complex ultrastructure of the eggshell. So you have different molecular actors that play a role. I will not give you all the detail of this molecular actor, but you have to know that you will have some molecular actors that will make the police, I will say. They will control the entry, what is allowed to enter, what is not allowed to enter into the cells. And you have some molecular actors that act as a transport and logistic that allow to the transfer of calcium or different ions, different molecules which are necessary into the uterine cells. And when you have all the necessary molecules in the forming milieu, you have the builders, which are another category of molecular actors. So in our lab, we have studied all these actors. Just to give you a more scientific diagram, you have here the blood, you have the cells, you have the uterine fluid where the calcification takes place, and you have the forming eggshell, which is here. And first, you can have a passive diffusion of CO2 in uterine fluid in the cell, and this CO2 will be directly transformed in bicarbonate by carbonic anhydrase present in the cell and uterine fluid. And you have what is the most important, the transcellular transfer of calcium, with some specific actor, SLC mainly, that allows the penetration of bicarbonate, but also some other SLC that allows the extrusion of bicarbonate from the cell into the forming milieu. And the same for the calcium. You have TRPV that allows the calcium entry in the cells. You have calbinding that play an important role to transfer the calcium and maintain at a very low level the internal calcium concentration. And finally, if you have a storage in a reticulum endoplasmic foam cells and some anion exchanger and that allows the extrusion of calcium from the cell. So you have sufficient of calcium and carbonate now to produce the eggshell. But there is another important factor that's come in the game. This is what we call the ACC, amorphous calcium carbonate. It's not possible in the normal pressure to make some calcium ion to a crystalline calcite. Calcite, this is a very strong crystal of calcium carbonate. This is a marble, for example, in geological earth. So we need to have some specific structure of calcium that will reduce the level of energy to obtain the calcium crystal. And this is the amorphous calcium carbonate that play this role. And it will be transferred in vesicle to be stabilized and to help to the fabric of the eggshell. Okay, and finally, just to show you that is under the dependency of estrogen receptor and vitamin D receptor on the nucleus. Okay, this is a general scheme describing the different timing of the eggshell formation, the different steps. And to summarize this part, I will say that the eggshell is composed of 95% of calcium carbonate, this is calcite, and a little content, mass of content of organic matrix proteins and proteoglycan, 3.5%. But this is the interaction of both organic matrix, mineral phase, and organic phase that give to the eggshell mechanical property. And the mechanical property will depend on the quantity of eggshell, but also of the ultrastructure of the eggshell. So how it is fabric, how it is organized. It will be responsible for 40% of the mechanical properties of the eggshell. So what have been done in the past, and we have contributed a lot in our lab, is to identify the organic matrix proteins. In 20 years ago, I knew only 3 or 4 organic matrix proteins in the shell. Now we know that there is more than 900 proteins in the shell. And these organic matrix are important to stabilize the amorphous calcium carbonate, to control the shape of the calcium carbonate and the size of the crystals. Imagine, this is a theoretical example just for understanding. Imagine you have an original crystal in this rectangular shape. If you add some calcium and carbonate in the forming milieu, you will have the same shape of the crystal. It will be larger, but the shape will not change. But in presence of organic matrix, they can absorb on specific phase of the crystal. And the consequences is that this growth, the B-axis, is inhibited. And finally, you will have a final shape of the crystal that is modified, that is elongated. This is what we call the anisotropic growth. In the real life of the chicken, which is what looks like a calcite crystal in absence of additive, organic material, and this is the shape of the chicken calcite crystal that is modified by the organic matrix. And how it is important? It's important because it's allowed to establish a preferred crystal orientation in the eggshell. Let me show you that. You have the first crystal, which are elongated due to the matrix protein, that are crystallized around the mammillary knobs in all directions. And you can see in the second step that, for example, this crystal cannot grow because there is a competition with adjacent crystals. And finally, if you go to the external part of the eggshell, all the crystals are perpendicular. And a big advantage in the point of view of evolution is if you put a stress on the surface of the shell, all the crystals will push together and will be like the stone in the casserole. It will be very strong and solid. But from the outside, when the young chick wants to go outside, it's really easy to open it. So very strong structure from the external shock, very easy to open from the internal part of the structure. So I told you that it was what is important. This is a quantity that defines the mass of the eggshell, but also the control of shell calcification that defines the ultrastructure of the fabric. We know that the mass contributes to 60% of the total solidity of the shell, and the fabric contributes to 40% of the solidity of the eggshell. So how we can use this data to improve the eggshell quality? The mass have been mainly used in the past by nutrition, genetic, environment, light programs, and the potential gains are now slight. But the ultrastructure is less studied, and potential gains are really elevated. We can use some polymorphism of gene coding matrix protein, which are under control on vitamin D, to get some genetic and nutrition interaction and not intercation, like I have written, and to improve the eggshell. And it will be my conclusion to say that in the last 20 years, a lot of scientific programs were performed to identify thousands of molecular actors involved in the eggshell mineralization. This molecular player show polymorphism associated with differences in shell quality. I do not have give you some data about it, but I can if you like. And it's allow us to go to a genomic selection to obtain some favorable haplotypes with strong eggshell mechanical property. Additionally, this molecular network shows differences in abundance according to vitamin D status. So it allows us to go to precision vitamin feeding of the layer to reinforce the quality of the shell. Both pathways used in conjunction help us to give us some shell with enhanced mechanical property even during prolonged production cycles, and it ensures the health and the absence of food outborne for humans. So I thank you very much, and to end, I would like to thank the people of my team and from the unit, and also Alejandro Rodriguez Navarro from the University of Granada, which I am collaborating for that, and Max Inke. This is collaborators that I used to work with them till 1993 now. Okay, thank you very much, and if you have questions, I'm ready to answer.
Thank you, Dr. Joel. If anyone have questions, please. Yes.
Thank you indeed. Thank you, Dr. Joel, for joining us in this Congress. I have a more applied question. If, starting from this research of yours on the eggshell formation and crystallography, did you find any extension to a cross-disciplinary technological transfer? I mean, starting from the improvement of the shell for our safety. One of your friends in crystallography, did you find any others idea of applying this, like nanomaterials or something? If you could detail, please. Thank you.
Yeah, of course. The fabric of the eggshell, the crystals which are organized in the eggshell are nanocrystals, really well organized, like we have in nanomaterial science. So what we have discovered, we have some program with German people of the Max Planck Institute, for example, of material sciences, because what we know on the eggshell can be useful to determine some additive that will be used for the material sciences. And there is a lot of department of material science who want to mimic what is living to living organisms. And finally, from biomineralization on natural nanostructure, this is in the layer and the eggshell formation where we have the most abundant data. So many of people are looking of the organic material that we have identified to know how they could exploit in the future to perform some new additives for synthetic nanocrystal in the future.
Thank you.
Thank you very much. Thank you.
Other questions? No. So, thank you very much for your presentation. And now I take the floor to Mr. Professor Mihai Datcu to present his lecture.
Good morning. Dear Professor Miro, thank you very much for the invitation and for organizing this excellent event. I will start the presentation with a very short movie without sound and without words. Sound. everyone is aware about the effect of the climate change. So that's no secret today. And actually, for the observation of the Earth, today we have a technology, the only technology which is able to have a global and a continuous observation, the satellite observation. We have now 50 years of history of satellite images, which are observing the three main factors for the climate change. The ice melting, that is a synthetic aperture radar from the TerraSARIC, German Aerospace Center satellite, observing the melt of a glacier, measuring how the glacier is melting and breaking in the ocean. The second important factor, so this is a map of the speed of the glacier flowing. And the second main important factor in the climate change, it is a forest. That is, again, a TerraSARIC image of the rainforest in Brazil. So deforestation is one of the factors which is preoccupying us. So in red and in blue, there are areas in the rainforest which have been deforested, and there are crops, sometimes collected, sometimes legal, sometimes illegal crops. And the third main factor in climate change is us, the human activity. I'll continue with another short movie which actually shows the motivation of the activities in Earth observation. So which are the challenges in using the satellite information for monitoring the Earth. I'm letting now the movie run.
Information being lost because data is being collected that can never be analyzed. The German satellite data archive near Munich contains vast amounts of valuable data collected over the past 30 years. The archive can store more than 50 petabytes of data on magnetic cassette tapes. It may be that these tapes hold the answers to questions that won't be posed until sometime in the future. But when that day comes, will we be able to access the data? The more data we save for later, the greater the risk of losing information forever. Even the best storage media can age and become obsolete. But maybe there's a way to master the deluge of archived images and virtual reality. Researchers are mining the satellite data archive, hoping to make its information more readily accessible. As if the archive were a library with open books lying before them, the scientists can pick out an image, dive into it as it were, and check to see whether it shows structures similar to those on other images. The computer works like an automatic sorting facility. It searches the images for components like vegetation, farmland, bridges, and water. The computer sorts the images into stacks and categorizes them by their content.
At the present, they probably store in archives more than 10 to 20 millions of satellite images acquired after the 80s year. With a new era of the Sentinel satellites of the European Space Agency, the data volume per day will reach the level of 10 terabytes. This is our data rate. So we are going to reach soon thousands of petabytes of data, which is a large volume of data, and all the research community is putting huge efforts in dealing with information extraction.
Soon we will no longer be talking about millions or billions of images, but trillions. And then it will be more important than ever to uncover meaningful information hidden in the mountains of data.
So in summary, we have the challenges of climate change adaptation. We have a lot of data, big data from satellite observation, which contains information in support of the measure for adaptation. However, to exploit the information in this big data, we need technology, like artificial intelligence. Artificial intelligence, as in this movie, we can dive in big data, in terabytes of data, hundreds of terabytes of data. And when the PhD was looking to an image of the coastline, the archive turned in front of him all images of the coastline. When he was looking to ice patterns, the archive turned in front of him all the ice patterns. So that is a transformation of big digital data in meaningful information in such a way that people, scientists, industry can use the content of the data. So this was a motivation of applying Earth observation for climate change and using advanced artificial intelligence. I'd like to acknowledge my team at Polytechnica Bucharest for the work. Part of the result presented today, I would like to acknowledge my team at the German Aerospace Center and Munich Aerospace, and at CNAM, Conservatoire National d'Art Métier, under the Chaire Internationale de Recherche Blaise Pascal. So today, together with my colleagues at Polytechnica and colleagues at National Institute of Earth Physics and GreenSoft, I will make a short overview of a project we started, the Competence Center for Climate Change, developing digital twinners to forecast and societal redressment. So Earth is changing. So we have pressure on resources such as fresh water and food, stress on land and marine ecosystem, environment pollution, impact on health and biodiversity. The global warming, it has an effect on the rising the sea level, increasing the acidification of ocean, and definitely extreme events, climate events, weather like floods, heat, or drought. So today, after a period in the last 10 years when we mastered big data, we mastered the technology to collect, store, transmit, process big data. After the last 5 years when artificial intelligence was developing a lot, we now have the tools to access information and big data. Today we are at the edge of a new technology, digital twinners. So digital twinners should provide capabilities to visualize, monitor, forecast natural human activity on the planet in support of sustainable development of European and Green Deal program. So digital twinners is a technology which is combining, integrating big data, artificial intelligence, modeling, physical models, mathematical models, visualization, and doing prediction, forecast, and implementing what-if scenarios. It's a new technology emerging. It's not only observation, but Earth observation is a particular field. Now, being this challenging and these requirements, at a national level, since last year, we started a project to establish a competence center for climate change adaptation. It is a project under the National Program of Redressment and Resilience, in which were involved partners, university and industry. We are working together with the colleagues, with Professor Miron at University of Science in Iași, with a company, industry, GreenSoft in Iași, with the Romanian Space Agency and Multidimension company in Bucharest, with the colleagues at National Institute of Earth Physics and GreenSoft, a company accompanying them, and the University of Vilnius in Constanța for marine application. So we are in the beginning 10 partners, and we would like to extend the competence center at a national level. Definitely climate change adaptation is a very broad field. We focus on the following problematic. Climate changes are at scale of global scale, tens of thousands of kilometers. However, adaptation measure should be applied for areas of 10 meters, 10 kilometers. Climate change, it's observed for tens, hundreds of years. Adaptation measures should be at the scale of human activity, days, months. So we focus on adaptation measure and the scale of human activity, so scaling in high resolution in space and time. So the Competence Center will be an organization which will operate a national network of excellence, including our partners in the project and the stakeholder. We have contacts with a main European agency, Environment European Space Agency. However, to operate the Competence Center, we develop digital media. The digital media, it's a federated system of digital twins. We have a digital twin which is extracting the information, physical, biochemical information from satellite images. And this information will be used by specialized digital twins on One Health, dealing with the risk of new disease carried by mosquito and ticks. And one of the major effects of climate change, biodiversity, extreme events, and water quality, the Black Sea and the Limitrof lakes. Now, to implement this technology, these digital twins, we need to rely on artificial intelligence. And we need a different artificial intelligence. So most of the work in artificial intelligence, it's for text, multimedia, video, pictures. Here we deal with sensor data, sensor carrying information. And we need a new way of a new approach. In this slide, I make a brief introduction in remote sensing. So information and remote sensing satellites are presented here on three axes in a three-dimensional space. One direction, one axis is a pattern, information and pattern and objects. The other axis, it is the physical parameter. And the other axis is geometrical parameters. So we have sensor in space satellites which are obtaining very high resolution. We can observe airplanes, cars, roads, ships, and containers. Also, we can count how many containers are on the ship. We have also satellite sensors which are extracting physical parameters. Sometimes we have a sensor which is measuring a single physical parameter, ozone concentration, for instance. And we have another category of sensors in space which are measuring geometrical properties, like the terrain height, surface model, digital elevation model, or the subsidence, measurements which are done at millimeter precision for very large areas. And definitely we have sensors which are in between, sensors with very high resolution, measuring, recognizing patterns, objects, but also carrying information, physical information like vegetation, water quality, and or temperature. So satellite images are really not images. And I give only two examples. This is a satellite image for Copernicus Sentinel-2, a sensor, multispectral, with 13 spectral bands, 3 of them invisible, from which you can obtain a color image of the land, and other bands in infrared. So that is Dobroja. We have Constanza, the Black Sea, the lakes on the Black Sea. We see agriculture, bare land, Danube, a cloud, and the shadow of the cloud. That is an infrared band. It's the same image. It's not another. It's just one band of the same image. What you observe, the cloud disappear. Why? Because infrared is a wavelength larger than the particle of the cloud, and there is a diffraction process. So it's a physical process. So in here we deal beyond photography, beyond visual perception. We measure physical properties. However, the cloud disappeared because it's transparent due to diffraction, but the shadow of the cloud is still here. Why? Because infrared is measuring temperature. It's like putting the hand on ground. So the temperature in the shadow of the ground is still visible. Physical information. Second, synthetic aperture radar. So we carry radars installed on satellites. They transmit pulses like the police radar, and the echoes are coming back to the satellite. We transform these echoes in an image. It's not an image. It's a record in 2 dimensions of a radar signal. And that is a railway in California. Due to the relief, there is a loop. Railway goes in the tunnel and continue. And that is a synthetic aperture radar. We see the railway goes down in the tunnel and continues. Where is the train? The train is here. Why? Because the synthetic aperture radar is measuring physical parameters, the Doppler frequency. Microwaves are reflecting on the train is moving, and is changing the reflection, the echo frequency, Doppler effect. It's clear that these are not images. These are records of physical parameters. And that's why we need a new kind of artificial intelligence which should reflect and implement. And we developed a new concept, a concept which is based on 2 main ideas. First, we have knowledge about physical processes. We know how the satellite instrument is working. We know how the microwave or infrared is reflected by different land covers and objects. And if we know this object, we have to embed in our algorithms these models. But the nature has a huge complexity, and we cannot model nature. And in here we have the advantages of artificial intelligence, learning from data. And it's a concept to have a loop in which we are using existing knowledge by physical, mathematical models, chemical models, which can be applied on our data. And for the part of high complexity interaction, of high complexity scenes with incredible number of parameters, we are using the set of artificial intelligence. We can train and learn now millions, tens of millions of parameters from the big data. And I give only a very short example. This is a deep learning. I mean, I don't want to make you afraid. It's a deep learning architecture which is different from computer vision or speech or text analysis. So in here we are learning from synthetic aperture radar. We are learning from the Fourier transform of the synthetic aperture radar, and we are learning from semantic. Semantic which is given by experts. And the output of such a network, it is like this. We have the Eiffel Tower, and that is a full polarimetric, so radar can transmit vertical, horizontal, and diagonal oscillation of the microwave. But these polarimetric sensors are expensive and very few. But we have a sensor which is a single polarimetry, which acquires continuously all the world and provides free and open data. That is Sentinel-1. And we are training a model from few expensive data of polarimetric, and we are predicting polarimetric signature. That is a virtual sensor. So we obtain a signature, an image if you want, from an unobserved spectrum of the microwave. And the second aspect, we make classification like an application sea ice, which is very important for climate, like a young ice, first old ice, glacier, iceberg. But we don't make only a classification. Each of the class, it's actually explained, because inside of the class, we understand the type of scattering, diagonal, dipole, Bragg, double reflection. So we have physical information, and we need methods to extract the physical information from our sensor. Another example, we have a LiDAR sensor, JETI, on the International Space Station. This sensor is measuring the canopy height, but the measurements are sparse, both in space and time. And what we are doing, we train deep learning on this sparse data, and we apply to Sentinel-2, optical multispectral sensor. Again, continuous observation, global, free data. And we generate from Sentinel-2 with this model, with this algorithm, the height of canopy. So yellow means higher height, blue, lower height. It's a virtual sensor. We transform an existing free sensor in a very valuable information, global and timely. We have another asset. After 50 years of remote sensing, each side of the Earth was observed many, many times. Only from Sentinels, we have on every side of the seat of the Earth thousands of hits. And these are satellite image time series. So today, Earth observation is not anymore one image. It's a history of an image. And that is Bucharest from 1984 to 2012, like a movie from space, where you can see the growth of the city. We can see the change in agriculture after 89. We can see development, construction of highways. We can see expansion of urban mixing with industrial. It's a huge value in satellite data time series. So these are dynamic objects. And from this dynamic data, we extract not only land cover, but we extract dynamic processes. We have a history from which we learn. But the satellites are coming and revisiting the same place on Earth only a few days, typically 10 days. And this is a challenge. For this monitoring of frequent observation, we need from satellite constellation of 100 satellites. I mean, if we want to have observation every 2 hours, we need 100 satellites. That is expensive. And in the cooperation with our colleagues at the Institute of Earth Physics, which are, they installed and operate a large network of sensors for seismic and acoustic, seismic, microwave, lightning sensor, and all these sensors are recording data on a continuous basis. there is a sample every second. And actually, our colleagues, they opened a disruptive solution which we are using the seismic data. Seismic data can have records on very large areas, like tens of thousands kilometers. And again, these sensors, they are collecting data all the time. A satellite is coming and revisiting it every 10 days. And my colleague prepared a live demo, and as any live demo requires technology.
Just a second, it's now there.
I monitor the activity. Mrs. Mihaela Gavrilă from Greensoft will continue the presentation. Live demo is a challenge.
Perfectly. I'm Mihaela Gavrilă and I represent Greensoft. In this project, we developed a platform where we can see the meteo events from collected from sensors. We can select a region. We can select typical sensors to see the information in real time. We can set the time period to see the events on the map, and also we can set the hour. No. Okay. So on the map, we can see the thematic layer we selected before, the storm. We can see the strike from I don't know how to use the laptop. I'm sorry. Escape. Help me. It's in work in progress, the platform, but I don't understand why I don't see the menu. If I try again and again, I don't see the menu, because I would like to apply the select analysis type of the layer and the thematic layer, and on the map we should have all the information bringing to data set. But I don't know why, because this Chrome, and I don't know why. I'm sorry.
It is emotion.
It's The link is can be accessed from the phone, and you can see individually the platform and the data collected informations. Let me try again.
Let it on the screen.
Let it. Thank you. I'm sorry.
No, thank you very much. Could you put it back on the screen? Mm-hmm. The link.
The link? Yes.
So, if I take one of the microphones. Mm-hmm. So again, it's a disruptive approach in which are combining measurements which are designed mainly for lithosphere. And these sensor networks that are operated in Romania, they have a visibility of 10,000 kilometers. And again, the records are done every 10 seconds, continuous. And we want now to fuse this data or satellite data and extract physical parameters from indirect observation and to extract information from all sources which exist. In addition to this, one of the challenge which was raised by our colleagues at the National Institute on Physical Atmosphere, it is to study the effect of the climate on the lithosphere aspect. It's working, of course.
Okay. We bring the data collected, as we said before, and in the same time we can see the heat map, where the density of the number of events in region we can see using the heat map, and of course the individual events from storm and strike. We still work on this platform, and we will bring more data, of course, and more sensors to see in real time how they can complete a phenomenon. This is in a public place of platform. We have also a private place where we can access it with the user and the password. Okay. And here we can see the map, but we have an administration place where we can manage data from sensors, data source, and event observation. Of course, we can manage the users, the metrics, the health configuration, and of course the logs of the platform. The sensors from data which we see before is only one for the moment. The data source, we can see the integration with the platform to collect, who collect from sensor directly, and event observation. All events presented before is here in this list. Thank you.
Thank you very much.
Thank you.
I thank you, Mihaela, and I thank you also Dr. Constantin Ionescu, the director of National Institute on Physics, who is present in the room. And the application, it's available also on mobile phones, on application. It's much more, it's closer to real time compared to any meteo. It's not meteo information. It's very precise, time and position, and it's dynamic. With this, I conclude the presentation, and thank you again.
Thank you, Professor. Question, please, from audience. Remarks? You have one. Okay.
Hello. What do you think about Copernicus? Copernicus, you know, the European space program.
Yes, we are, in the digital twin, we are using all the data from Copernicus. We started with Sentinel-1, Sentinel-2. We work on Sentinel-3. And also we are using the products which are provided recently, which are very rich information from the Copernicus services, land, ocean, atmosphere. But we focus on real time, as close as real time, because the services are actually extracting information which from it's integrated for a period of time. And if we want to have observation in real time, high frequency in time observation, we need to go to the satellite and to the data which are coming from the networks. And so the goal is to go to this very fine scale of time. To go from scale of time of hundreds of years, tens of years, 5 of years. I give an example, Corin land cover, which is one of the Copernicus product. It is renewed every 4 or 5 years. And Corin land cover contains land cover classes. If something is changing from a day to the other, we don't see in Corin land cover. We can use it as a support to understand, to have a context. But the real-time information is coming first satellite observation a few days, and later we want to focus in time to have a zoom, a loop on time, which goes to minutes, hours, and this is coming from sensor which are operated by the Institute of Physical Affairs.
Thank you. Thank you, Professor. I have just a comment because this time passed quick. We have 2 p.m. workshop dedicated to vector-borne diseases in which all team of this DTE climate will participate with papers, and we'll discuss in this workshop how we can use AI just to involve satellites to analyze microhabitats, because now we can use this one just to have different measurement with a high sensitivity, but we try to have, in fact, finally, after this project, the possibility to analyze microhabitats where are living arthropods and their life cycles, just to have finally data from this development of insects and ticks, for example, and to know better about this climate change dedicated to move and change their life cycle. It's important, just emerging diseases to human and animals in the One Health context will be well defined. Professor, thank you very much, and we expect all 2 p.m. in our web disease workshop. If you are not time to participate directly in TPPA Amphitheater, please follow the link. You will have the link post on the Congress site, or if not, to your moderators to sections. Thank you very much. And now, thank you again, Professor.
Thank you. Thank you very much.
Shall we continue with the fifth presentation, the stereotypes and the influence on cognitive performance? Sharman Moroshan.
Thank you very much. Thank you very much, Professor Datko, because you, in a way, you introduce my topic. My topic will not be with artificial intelligence. My topic will be linked to our brain. That is the name of my topic, the stereotypes and the influence of the cognitive performance. Actually, one of my job is to represent Sorbonne University to LERU. LERU is a European League of Research Universities, which means the biggest 24 research university in Europe. And a few months ago, because we have different type of meeting, different type of subject, each person represents university in their expertise, of course. And one of the topics during the meeting a few months ago was stereotypes linked to the universities, academic, and the influence of different stereotypes in our activity, how we evaluate our students, for example, how we recruit the new researcher, the new director of research, etc. And this is my topic. What about the cognitive aspects of stereotypes? They focus on how our mind uses the mental shortcuts to process the information about the others, and this is very fast, which is good for our brain. And stereotypes, all types of stereotypes, play a significant role in how we process the social information. And the social stereotypes are often, unfortunately, very caricatural. And they fulfill two functions. One, it simplifies our environment in which we live. And the second one, it's, let's say, it's a little bit more difficult, but in one word, they maintain a social hierarchy between two groups, dominate and dominated. The definition in psychology of these social stereotypes is this one. It's a set of shared beliefs about the personal characteristic. Usually it's personality trait. Competence, sometimes unfortunately it's incompetence, but also the behavior of a group of people. According or based on the content of stereotypes, we, gentlemen, all here, we are competitive, we are daring, we are rational, we are good in mathematics, but we are bad in reading, managing interpersonal relationship and emotional situation. And you ladies, without exception, you are sensitive, emotional, you are good in writing, but you are bad in mathematics, logics, spatial orientation. How many times we don't hear, traffic? Who is driving this car? It's a woman. And leadership, which means you cannot be, let's say, the true. You cannot be, the president of the country or, a leadership. But this is according to the social stereotypes. And I would like to share with you two types of research. On your left, you have this research called so-called threat effect of stereotypes. And this can have an effect on self-evaluation, let's say my self-evaluation, and the learning and performance, my performance or your performance, if I evaluated you. And on the other hand, you have another research, and the name is carrier of stereotypes, which means all the persons, men, humans, social conditions not important, who they believe they exist, these kind of stereotypes. And this, unfortunately, can have a consequence and the effect on perception on how we judge somebody when I want to recruit, for example. And let's give you some example. This kind of two research, they have a common point. And this common point, neurological, scientific point of view in our brain, the name is automatic activation of stereotypes. And so let's make together a small test. It's less than 20 seconds. I will project on this screen a stimulus. This is a classical test used in psychology, so it's nothing special for the memory, for our memory. And so I will project a stimulus. Stimulus will be a color. The classical color red, green, blue. And when you will see the color, please, all of you, loud, just name the color. Okay? Ready? But be careful, we'll be very fast. Let's start. Thank you very much. I think it's because you are hungry, but anyway. So I heard you are very shy. But anyway, I heard some errors. So why? Don't worry. You don't have some, neurological disorder. You are, scientific point, you are normal lectors. Why? Because you learn to read. And you have somewhere in our brain, somewhere in your memory, and you automatically, you activated this one. It's simple. It's nothing. What is this? Automatism. It's a spontaneous. It's a rapid responses without a big effort from, our brain. But this automatic response can lead to errors. You did errors. You don't name the color. You read the word. The good news is we can, it's not very simple, but we can control this kind of automatism. But if I control this cognitive, have a cost for my brain. I have to focus. I have to find, okay, it's not the word, it's etc. This is millisecond, but it's an effort for our brain. And of course, another good news, if I stop this stereotype, this automatism, this does not remove from your memory, because we still know to read. And let's take 2-3 examples about the effect of stereotype threats. This is according to a researcher. It's an American social psychologist, professor at Stanford University. So, effects. Reduce performance of individual in situation where they fear to confirm. I'm fear to confirm that I'm not good in math. In my eyes, or in your eyes, it is negative stereotype and targeting their group a given area of ability. Example. Effect of stereotype threat in women. Ladies. So this is a classical mathematical equation. To be honest, it's not very difficult. But anyway, let's say this group, you women and men, and I will give you standard information. I want to see how good you are in math. That's all. This is the result. The men are better. This group, I give you the standard information and I add. Don't worry, be careful, I will never compare your results in mathematics. And you ladies are better, just because I influence some stereotypes in your brain, in our brain. Let's say the gentlemen. And the researcher taken, a group of gentlemen, let's say, without problem, caricatural problem in their life. Why? European, means white, rich, graduated of Stanford University, which is a big university, right, in the United States, a very famous one. And I wanted to say, okay, this standard is a very skilled in mathematics. They succeed 9.6% to 10. It's a good result. And this group, I said, be careful. I will compare your test with Asian students. And the United States reason a stereotype, according to this stereotype, the Asian students are better than European students. And the gentleman lose 3 points, more than 3 points, which is significant. Statistically, it's very significant, just because I said. So any of us individual can fall victims to the stereotype threatening effect if the situation leads to it. And the consequence can be how? It's additional stress. I'm good or not I'm good. And the cognitive overload, underestimation, and performance below actual stress. We are in a university, and we receive a lot of, CVs. And if I have this profile, I want to hire somebody competitive, ambitious, and assertive. Gentlemen it's a little competent. And exactly the same for the ladies. It's competent, but it's a little bit hostile. It may be cold, I don't know. But if I have another profile, I need somebody modestly, kindness, empathy. The gentlemen are not very good evaluated. The ladies are very good evaluated. And the jury, trust me, it's more than 1,000 jury different women's and men's. And the last example, and this influence of gender stereotypes on CV. So you receive a CV, we receive a lot of CVs every day, and on the left, you have a jury formed by ladies and by men's, excuse me. And everybody, they evaluated better the CV of men's. And the other side, it's ladies, they evaluated in the same way. And even worse, I'm sorry. I mean, the ladies propose a salary lower than the men's propose. So it's very important, this is how can we fight with this kind of stereotype? And there is a lot. Be aware that they exist. Secondly, knowing what we ourselves can be influenced by this. Understanding how it's influenced and knowing how it's possible to control this. In conclusion, this kind of stereotype are present in all individuals, women, men, young or old. So if you hear, dear students, don't think if you are young, you don't have this kind of stereotypes. You have. Old, don't think you are old, no. So risk. For example, we have a problem in the diagnosis of Alzheimer's diseases because it's a test, and because the old people, when they arrive in the hospital, it's a false diagnosis because I'm old, so maybe I have Alzheimer's. So all this knowledge, scientific point of view, is important for our life. And it's completely normal and common, but the consequence can be not nice, can be harmful. And one example of publicity of environment, how environment can educate us or our kids, our students. I found this 2 weeks ago it's a very known brand in France, Tartine Chocolat. So they decided to do like this. So Monday and Tuesday, the girls are artists, but the boys are sportive and the genius in mathematics that I gave you the response. And I remember, I don't know if you remember, and I have a short 2 minutes video and I'm done. A very nice publicity for the Nike brand. And for the Nike brand was good because it was exactly to fight against different type of stereotyping. One of the brand of the publicity was play like a girl, run like a girl, and was great. And I found a podcast, what are the opinion of the young boys and the young girls about fight like a girl, kick like a girl, run like a girl, and play like a girl.
Hi, Erin.
Hi.
Okay, so I'm going to just give you some actions to do. Just do the first thing that comes to mind. Show me what it looks like to run like a girl.
My hair.
Show me what it looks like to fight like a girl. Now throw like a girl.
Aww. My name is Dakota, and I'm 10 years old.
Show me what it looks like to run like a girl. Throw like a girl. Fight like a girl. What does it mean to you when I say run like a girl?
It means run fast as you can.
So do you think you just insulted your sister?
No. I mean, yeah, insulted girls, but not my sister.
Is like a girl a good thing?
actually don't know if it's a bad thing or a good thing. It sounds like a bad thing. It sounds like you're trying to humiliate someone.
So when they're in that vulnerable time between 10 and 12, how do you think it affects them when somebody uses like a girl as an insult?
I think it definitely drops their self-confidence and really puts them down.
1 minute
because
left.
during that time, they're already trying to figure themselves out. And when somebody says you hit like a girl, it's like, well, what does that mean? Because they think they're a strong person. It's kind of like telling them that they're weak and they're not as good as them.
And what advice do you have to young girls who are told they run like a girl, kick like a girl, hit like a girl, swing like a girl?
Keep doing it because it's working. If somebody else says that running like a girl or kicking like a girl or shooting like a girl is something that you shouldn't be doing, that's their problem. Because if you're still scoring and you're still getting to the ball on time and you're still being first, you're doing it right. Doesn't matter what they say. I mean, yes, I kick like a girl and I swim like a girl and I walk like a girl and I wake up in the morning like a girl because I am a girl. And that is not something that I should be ashamed of. So I'm going to do it anyway. That's what they should do.
If I asked you to run like a girl now, would you do it differently?
I would run like myself.
Would you like?
Thank you. No questions, please, because it's not veterinary science, so, and you are hungry. Anyway, if you have a question, if I can, I can.
Thank you, Sherry. Question, please. No one.
Thank you very
Girls,
much.
not from ladies, not from
Thank you.
men. So we are late. Our programs mean that just to 2 p.m. we have a brunch in front of our hall, and after you are free till 2 p.m. you will follow each one the sections who are posted here in Congress if you want to read it. Thank you for this first meeting, and we expect you don't forget that have in your bag your voucher to be tonight at 7 o'clock to La Castel restaurant. And for going there, we have buses to 6:45 posted in our court just to transfer you to the hotel. Yes? So don't forget to have the voucher in your bag with you. Thank you very much and have a good afternoon.
No,